Well, thank you very much to Karin and Nicole for excellent lectures. Karin for covering in detail the physiology of mRNA vaccines, the general place, the context in which they're developed, and the enormous numbers of possibilities that they may address. To Nicole for introducing us to the enormous spectrum of opportunities with gene therapies and other innovative therapies. I know that Nicole is under a little bit of time pressure, so I've got several questions here, and I was going to begin with her questions. Yeah, there's a nice broad question for you here, Nicole. In your opinion, which is the most important challenge that FDA is facing regarding regulation of gene therapies? What are your opinions there?
Yeah, thank you for that question. It's an extremely important question. So, gene therapies, they provide a lot of opportunity to address significant unmet need in rare disease. And so I think one of the challenges is really the balance of getting therapies to patients that need them as in an expedited fashion as possible, while also, maintaining all of our standards that are quite necessary in terms of safety and efficacy. So one of the challenges I would say is because we have such targeted and directed therapies, that we can get things to patients sooner, but there's significant heterogeneity within, in particular, the rare disease space. So we really need to be able to follow patients, after approval for longer term studies, and really getting that commitment and truly understanding how to do long-term evaluation in the post-market setting effectively, so that we can continue to monitor and to continue to see how these therapies work over time, while also maintaining our commitment to getting things to patients as soon as possible. So a lot of challenge, but a lot of opportunity and a lot of exciting products and a lot of exciting work right now in the field.
Thanks very much. I think that's an excellent overview of the challenges. And I would imagine from my drug perspective, another challenge is that we just don't have that many patients to work with. So I think we're certainly sympathetic to the environment in which you're working under, with really limited number of patients and novel technologies. I had another question for you, Nicole, which was, in the Office of Therapeutic Products, is CAR T included? Perhaps you can just expand a little bit on what your approach is to CAR T.
Yeah, so we do have CAR T cell therapies within the Office of Therapeutic Products. That's where they are regulated. And I will say that's also a really exciting field. The CAR T space, as many know, has really expanded in recent years to not only include malignant conditions, but non-malignant conditions as well, and provide a lot of hope in a lot of areas that are really in need of more targeted therapies. So yes, they are in our office, and we do see quite a few INDs in this space that are very across the spectrum.
Great. Thanks very much, Nicole. I'm going to turn to Karin now with a couple of questions. And there's one question which is broad and I think makes a good beginning to the discussion. Karin, the question is, why is it hard to make vaccines for some conditions?
Thank you. You're right. It's a great question. Many factors, really, and one of the great examples is HIV. Why after so many years we don't have a vaccine for HIV? And it depends on the biology of the viruses. In this case, HIV, you would really need to prevent infection, not only disease, and that's very hard to do. It also depends on whether we understand the virus science enough, or maybe if a clinical trial is feasible, is if you have a reasonable clinical endpoint that you can measure the effect of a vaccine. So there are many, many reasons why there are some diseases that don't have vaccines, even though they have a great burden, from scientific all the way to practical reasons.
Thanks. I think an excellent answer to a question that faces all of us in the medical community. So a couple more questions for you, a lot of interest here. One question is that vaccine typically requires assessment of biological potency at release and for stability. And the individual is asking, how is this performed for mRNA vaccines?
Another great question. Very good audience today. So that doesn't depend on the platform. Usually potency depends on the disease and the pathogen that you're studying. So your potency assay, whether it is an animal model or immune assay or whatever else that you're going to do, depends on, or a correlate on what pathogen you have that vaccine against. So that's going to determine the potency assay that you're going to use, which is, can be used for mRNA as well.
Great, thanks for that. And keeping you in the hot seat here, there was a technical question which perhaps you can answer. Were pseudouridines or other modifications recognized as reactogenic causes of adverse events during the development of the mRNA vaccines for SARS-CoV-2?
Excellent question, and sorry I did not clarify. No, all that science was done years before. We were very lucky that it was all done years before the pandemic. And of course, mRNA is used for many other targets. One of the most important ones is cancer. So there was a lot of data accumulated and a lot of science behind pseudouridines way before the SARS-CoV-2 pandemic.
Thanks very much. Nicole, if you're still on, there's a question for you. For gene therapy, how much of a barrier is pre-existing immunogenicity?
Yes, so immunogenicity is something, as someone astutely has made that observation, is something that we really do consider and think through a lot. And in really some of the early phase gene therapy trials, we had a lot of lessons learned in terms of immunogenicity. So in the gene therapy space, in general, a lot of sponsors and companies and even academic investigators are addressing these potential immunogenicity issues to the transgene, in particular, with immune medications and other things around the administration of the gene therapies for a given amount of time to minimize the immunogenicity risk. So those risks are, as you point out, those are things that we consider in the development of trials and also in the development in this space. And we do have ways to try and mitigate those risks and then also to follow patients should this occur. So it is something that a lot of consideration is given to. I wouldn't say it's a barrier at this point, because a lot of people are addressing it in a way that does help with safety.
Thanks very much. A very good answer to a tricky question. I guess we have to be as creative as we can to make a question for Karin, which is close to my heart as an infectious disease doctor. How can mRNA vaccine technology contribute to the fight against antimicrobial resistance? And what potential does it have in preventing infections caused by drug-resistant bacteria? Any thoughts there, Karin?
Yeah, that's a tricky question. So mRNA, I mean, it might have a lot of different approaches. The one I know about, because that's not really my area of expertise, is I know we're trying to figure out how to deliver monoclonal antibodies through mRNA, which is being studied in several areas. And that, I imagine, will be an approach that could be used for AMR, but I'm sure others, maybe even Nicole, has more information about that. But it's a great question.
Thanks for that. Karin, I had another one for you, and I think we're approaching sort of the end of the session. In your presentation, you've mentioned multiple times that mRNA vaccines have been successful. Are there no safety concerns for mRNA vaccines, especially with COVID mRNA vaccines? So this is again about safety, which you did touch on in your talk. Any thoughts about that?
Yeah, and thanks for the opportunity to clarify, because yes, COVID vaccines were very successful. As you saw, some of the data, we prevented a lot of deaths and a lot of hospitalizations. But of course, any intervention, anything you take has a risk, and vaccines are not different. And we always evaluate the ratio of benefit risk. So in a way, that's the end point, evaluating the benefit against the risk. And of course, we continue to evaluate the safety of any vaccine, including mRNA vaccines and COVID vaccines, even post-marketing or post-authorization. So we are continuing today and in the future surveying safety events and informing the public of those events. So that's an ongoing activity.
Well, thank you very much. I think that brings us to the end of the session here. It's been a long and very engaging day. Again, very grateful to our speakers from the Center for Biologics on two areas of major interest. And at this point, we'll be closing for the day. I just wanted to let everyone know that tomorrow we are shifting phases, and we'll be talking primarily about investigator responsibilities and about FDA and our regulatory role as to ensure the quality of clinical trials and the data we receive. So we very much hope that you'll be able to join us for tomorrow for these very sort of practical aspects to clinical trials and investigator responsibilities.